Okay, maybe
All right.
should we get started? Maybe most of the people here.
Yeah, please. Yeah.
Okay. Yeah, it's a great pleasure to introduce Professor Eric Shea-Brown. Actually, Eric was also a lecturer last year, so we're really grateful that he's back. So as you probably saw from the webpage, Eric started his career in engineering physics at UC Berkeley, but then he completed his PhD in Princeton in applied and computational mathematics. So it's really really thoroughly quantitative backgrounds. He was at Princeton. He was advised by Professor Phil Holmes and Jonathan Cohen. And he did his postdoctoral training with Professor John Renzo at NYU on mathematical models in cognitive neuroscience and also in dynamics of neural circuits. So you can see what Ravas talked about, that the different levels of modeling. And today, in this lecture and also in the more advanced lecture later, Eric will talk to us about a really important topic in neural coding. So basically, as you know now that we can record from many, many cells simultaneously. So it's really how to make sense of it. Are they independent? Because if they're independent, they can code a lot of information. Or are they correlated? Are they correlated in what way? What are the intricate plays? I think this is a really probably, I think, the most important topic right now in neuroscience. So let's welcome Eric.
Great, thank you so much. It's a real privilege for me to get to be part of your summer school. And it's a real privilege to be connected to colleagues around the world, new colleagues and old colleagues. Some of you I've known for my whole career, and for some of you, this is a new acquaintance. So thank you very much. It's an honor and honor to be with you and sort of the meaning of it all. And please interrupt. I'll keep an eye on the time. We stop at 8, well, whatever time zone we're in, we stop at something 45, right? Is that correct?
Yeah, yes.
Okay, great. And we'll take a little break. So I'll keep an eye on the time, but as per however the summer school directors wish, I'm very pleased if people wish to just, I have my volume up loud, if you wish to just interrupt with any questions or any discussion, please do, and it's okay if we have to, with me, if we have to skip some of the slides, and for the sake of discussion. Okay, right, so the first thing to note here is that I'm going to share with you the work of many people and the ideas of many people. So those include Stefano Ratanatesi, Matt Farrell, and Marav Stern, and we all work together at the University of Washington. Colleagues from the Allen Institute for Brain Science, which is a fabulous nonprofit research institution in Seattle, just down the road from the University of Washington. So Michael Bison, Gabe Ocker, Guillaume Lajoie, who's in Montreal, Mattia Rigatti at IBM, and Madhu Advani, who at the time was in Boston at Harvard, Sophie Deneve at ENS, a colleague of Rava's, as well as David Dahman and Moritz Helias at ULIX. So there will be a bunch of topics. I'll review a little bit of the literature as well, but the results that I'll be sharing with you that have a more personal connection to are the work of all these amazing colleagues and collaborators. Okay. Great. So you've seen this, as I just learned very clearly from Rava and other colleagues, and in the clear introduction as well, that we're really going from a shift in terms of what we study as the neural code, from thinking about things one cell at a time. So perhaps, or will come upon this many times in your career, this famous work from the 60s by these authors, in which they showed different visual images, and they recorded what a single neuron did as that image was changed. And with just a single feature of an image, here an angle, it's possible to describe the neural code by plotting stuff, right? One thing on one axis here, the firing rate of a neuron, and something else on another axis, or at least you know what to plot on the dependent axis, because there's just one variable that you're measuring, at least in a small time bit. And there's been fantastic development at the level of single cells. Many of you perhaps have seen this paper as well, in which this question of what is being encoded by single neurons was expanded from stimuli that have a rather low dimension, right, like a single parameter here, to stimuli that have a high dimension, like different pixel maps or different images or even different concepts, and we see how single neurons respond there. But as we also have heard from our colleagues already today, these days in neural coding, what's on the vertical axis, right, the independent variable is no longer one thing, it's many. Even 5 years ago, we had these spectacular recordings at single cell resolution in which you see many neurons responding at once. Now, if we think about, and that's only expanded in other studies, which you know of, like these, in which there are not only many neurons recorded from cortex at once, but many neurons recorded from all sorts of areas, subcortical areas as well across the brain. Okay, so the point is that when we think about the neural code or what the brain is doing these days from the perspective of the data that's actually available, it looks something like this. Lots of variables responding at every moment in time. So that every moment in time, we have a giant vector. We'll call those vectors P. If we look at the ensemble of different vectors that are produced across time or across image sets, those ensembles then are points in very high dimensional space, in as many neurons as we have in the brain, or in at least as many neurons as we're able to record from. So I think the first question that we have is, how different is this picture about the neural code from what it is that we started with when we were plotting one or a couple of variables, when we're thinking about one neuron at a time. And that question is the one at the top of the screen, or it can be formulated in the way that we have it at the top of the screen, which is how many dimensions, how many different independent degrees of freedom does this whole group of cells have? So we can quantify that via an investigation of how the cells' activity are related to one another. As we also heard in the introduction, are they independent of one another so that you have as many degrees of freedom as you have cells, or are they somehow related to one another, in which case you'd have fewer dimensions or fewer degrees of freedom. Let me just pause there. Does this make any, is this sounding okay? Can you hear me? Is this making sense? Is this pacing okay? Too slow, too fast? Okay. Is it okay to refer to, when we look at some of the papers like the classical papers like from the 60s on neural coding, have people seen those before? Maybe could just one person unmute if you've seen them before, and maybe another couple people unmute and just say it if you haven't seen them before, just so I can get a sense. The papers of Hubel and Wiesel, for example, are those new papers to the group?
I think most of the people have some background in neuroscience. It's probably okay.
Yeah, so you've seen these things. Okay, that's great. So I have a good sense of the historical trajectory here. Okay, so here we go. So we're trying to talk about how correlated this activity is, or sorry, how far from independent the activity in different cells are, and that can be described in many different ways. But the basic idea is to look at responses from not just one neuron at a time, but at least two in this case. We can relate these to our vectors P here by remembering that different components of these vectors correspond to different individual neurons, and look at the responses of these different neurons as they're produced simultaneously over time, and produce some sort of quantity which tells us, are they showing independent activity or not. And this is the simplest way to do this, just by asking whether there are dependencies at linear order, so to compute a covariance matrix. And of course, there are more complicated ways of looking for departures from independence than this, but this is the place where most people in the literature focus. So let's compute a covariance matrix and look at, in particular, off-diagonal terms when I is not equal to j, and ask if this is non-zero or not, right? And if this is non-zero, then we know that there's some sort of dependence among the cells. But as we've also been talking about, we want to not just do this for a pair I and J, we want to do this for n squared pairs of cells. And so we end up with an entire matrix Cij. And the question is, what am I supposed to do with this? Let's get this. How do I summarize what I'm seeing in terms of the statistical dependence across all the cells in the population? And one way to do this is to say, well, I have a matrix of different covariances here. Let me look at the eigenvalues of this matrix of covariances. And what that ends up corresponding to is saying that if I'm going to do my best job of approximating the actual blob of neural responses, these Brown responses that actually happen with some sort of ellipsoid, an American football or a deflated Asian or European football that's stretched out in some way, what are the principal axes of that stretched football that best describes the blob of different neural responses? And so these things are the lambdas. All right, so there are a couple of different things that I could do with a Cij matrix. One thing is I could look at these lambdas somehow to try to describe the dimensionality of the responses through that lens, through the eigenvectors and eigenvalues. Another thing that's more typically done in many studies is to say, look, you're trying to tell me, are these cells correlated or not? Let's take all pairs Ij and average across them and come up with some number here, this average of C, and ask whether that is significantly away from 0 or not. Okay, so these are two ways in which people try to get at probing the dimensionality or the departure from independence when we measure many cells. Okay, so how does this go directly to dimensionality? Well, we've already described what these lambdas are. Let's define a quantity based on the lambdas, which is this dimensionality here. And this is basically taking these eigenvalues, the lengths of this distribution of points is spread out along the different eigenvectors here. Let's normalize them, and then let's combine them together in this curious quantity. So this is proposed by these authors in the literature as a good way of talking about dimensionality. So why is this even a good idea? To me, this looks like some sort of weird measure at this point that I can't really interpret. What is this really saying? Well, let's look at a couple of different cases. Let's say all the neural activity that I'm recording is just spread out across 3 dimensions, right? So it could be spread out in n dimensions and be a very complicated response, or it could be concentrated into a much smaller dimensional space. Let's say that's really what's happening. So in reality, we just have all my neural responses concentrated in 3 different directions. So the blob of responses really looks like this. Well, then what will the case be? So I know that I'll be extended in one direction. So one of my normalized eigenvalues here will be a third. So a third of the variance is in one direction, a third in another direction, a third in another direction. And then there will be no extent, right, of these responses. They won't be spread out in any of the other dimensions. So lambda tilde 4 through n will be 0. If I take these numbers and I just plug them into this formula here, I get 1 over a third squared, that's a ninth, plus a ninth, plus a ninth. Add them up and I get 1 over a third, which is 3. So I recover the dimensionality with which these points are actually spread out through this formula. And the same thing happens if I have another case like this. Let's say the points are more or less spread out in one direction with a little bit of extra fuzz or extra variability in that one direction. So lambda 1 tilde is almost all of the variability, 0.95, and you have a little bit of extra stretch in these 2 directions. Then you end up with a fractional dimensionality, and most of us would say that that's a reasonable way to actually describe this neural population. You'd say, well, I have n cells working together, but they really have a response as a unit that's a little bit more than one-dimensional in this case. Let me pause and take any questions here. Does this make sense what we're doing? We're taking the covariance matrix for all the neural data, we're processing it through the eigenvalues, and then we're doing this weird formula, but we like this formula because, at least in these limiting cases, this agrees with our intuition of what the dimensionality of the neural response really is, how large in terms of dimensions this cloud of responses that the brain is producing really is. So maybe at least someone, anybody who has a question first. Just wait for a couple of seconds. Unmute and share.
I actually have a brief question. So in the dimensionality, people use the square, right? Is there any special property to using the square versus other components? Yes.
Yeah, I think the only reason, as far as I can tell, is that if you take these limiting cases, then this will work. And I guess if we didn't take the square, then we would end up with a third, a third, a third, and we just sum them up in the denominator. You'd end up with 1/1, and you'd say, for example, right? And so that's a good question. I wish I could tell you better how it was that this was actually derived. But for me, I just, again, look at it from these different integer value cases and then note that this thing seems to interpolate between those cases in a sensible way. There must be a better answer than that. That's my best answer.
Thanks.
My best answer. It's a great question. Thank you. Yeah. Yeah, and there's interesting work that's trying to define better measures actually than this. Actually, I can make a more intelligent comment. So there is work that's on the archive now where the first authors are Brada, B-R-A-D-D-E, Brada, B-R-A-D-D-E, and Recanatesi, which is R-E-C-A-N-A-T-E-S-I. So those are the two first authors. And so what they do is they show that this measure actually relates to classical measures of dimensionality that are defined at much smaller scales, like the correlation dimension. So that's a better answer, is that if you take special cases of this, this relates to notions of dimensionality that are directly related to ideas of, for example, how will the number of points in a box scale with the size of the box, right? And that those type of quantities, again, coming from more classical notions of dimensionality, really do have to do with microscopic level, how many dimensions or how many degrees of freedom are there. So I think that relationship was not understood until a year ago, as far as I can tell. But this shows us that either we got lucky or there was somebody smarter than me who wrote this down in the first place, probably both. Okay, so that's a way of defining dimensionality that makes sense. Why are we even bothering to do this, right? So as per the excellent introduction, this is a fundamental question, right, about just what is the brain doing, right? And how different is the modern view of the neural code from a classical view that you construct one cell at a time. So it's kind of a fundamental question, but does this actually matter from the point of view of computation beyond just description? So what if the outcome was that the responses in the brain were really low dimensional? You have 100 billion neurons, right? But they really all sort of work together in such a way that if you look at their joint activity, the dimensionality is very low. Let's just say that was true, then what, right? Well, then you'd have many noisy variables working together to represent just a few coordinates. So that's a natural setting in which you'd expect denoising or canceling of any independent variability. At least you have many samples working together. You would realize that you don't need to read out that many cells in order to figure out all of the brain's information. But there's a much more profound implication as well. And that is that if you have a low dimensional representation like this, you don't need that many samples to understand everything that the brain is doing. Here's an example. Let's say that all neural variability, instead of being spread out in 3 dimensions here, is spread out along a plane like this. Then if you sample what the brain is doing as a downstream brain area, for example, that's trying to interpret what's happening in an upstream brain area, or as an experimentalist, you need 1 sample, another sample, and a third sample. And all of us know that 3 points then define a plane. And based on just what you know from those samples, you know everything about the ensemble of neural responses. That is useful because if you're trying to build a response that is going to work in general, that means that you can extrapolate from what you see from just a few examples and build a rule or build a way to read out brain activity that will continue to work. Now, what if we saw that the brain's responses were the opposite? They were extremely high dimensional. They were taking advantage of the fact that you have so many neurons in the brain to spread out responses at different points in time or to different stimuli all over the place. Well, there's a classical reason why this is a good idea. Let's imagine that we have two different categories of points, and this is something that we'll come back to in later lectures as well. So these points are neural responses when I'm talking in my visual cortex, when I'm talking to my colleague Sen. So Sen is looking very serious here. He's the X's. Okay, so here's Sen in the morning and here's Sen in the evening or whatever. But these are different groups of Sen. Sen on Zoom, Sen in a real conflict. And then these are the same ones. Lewis is more relaxed, he's nice and round. So this is Lewis when I actually meet with him in Beijing, or Lewis over Zoom, right? Two different versions of inputs that mean the same thing, colleague 1 or colleague 2. Okay, so let's say the brain produced a low dimensional representation of my colleagues like this. Then it's difficult to classify them. If I want to build some sort of a readout or a boundary that puts Sen on one side and Lewis on the other, it has to be some sort of a nonlinear function. But if instead the brain casts Lewis and Sen in very high dimensions, then in general, a linear operation, which is faster to learn and has fewer parameters, can separate any ensemble of points in any number of classes. And this is a well-known result that goes back far into machine learning and leads us to expect that maybe if what the brain is trying to do is to prepare images or prepare any sensory or cognitive states for easy separation downstream, so I can get all my Sens right and all my Lewis's right with a simple downstream readout, then maybe what it should do is produce these very high dimensional representations. And what's very interesting to think about is that there is a space in between where the representations are not so complicated that once I learn Sen's images up here, I will have no idea what Sen is doing on a different day. And so there may be a simple linear readout, but I need to experience Sen in all different conditions before I can generally recognize him, right? So it enables a good generalization. It's not too high dimensional, but it's also not too low dimensional, so that simple readouts can actually work in terms of interpreting the information. So something that combines the good features of low dimensions and high dimensions, we call this green dimension or a Goldilocks dimension. And many people have been thinking about these intermediate types of representations. So what I'm going to talk about next, given that I think we're motivated, hopefully we're motivated to think about dimensionality, again, not just as a fundamental description of what the brain is doing, but in terms of understanding the desiderata, the goals of its computation, and in particular, how it might satisfy and balance between the multiple goals that it has in which it seeks to be able to do general but powerful computation. Okay, so that's the motivation for this. It's a fascinating question also in data analysis, like we said, if we look at the literature. So this is, again, more perhaps in terms of describing what the brain is doing. So these are some highly recommended papers. This is Stringer, there's an R missing. But these are also questions that come upon us as we think about the mechanisms that actually produce these dynamics. And that's what I'm going to focus on for the rest of my time with you, both now and in the more advanced lecture, I guess. And in my simpler lecture, or more basic lecture, I've given this introduction, which I've just given. And I'm going to talk about a question which is, if not easy, very concrete, which is to say, well, you have a neural network. We're interested in the dimensionality of the activity that it produces. What is it about that network in terms of how it's connected that determines the answer, right? What is it about that connectivity that determines the dimensionality of the brain's activity? Okay, and when I'm with you again in a few days, we're going to come from a top-down perspective and say, well, what if that connectivity isn't just some arbitrary thing that we as mathematicians or theorists write down, but what if that connectivity is something that evolves through learning? Then in neural networks, what are the types of dimensionalities that actually, again, emerge from learning, and how do they relate to the task that the brain is solving? Okay, so we can see, I think that this is a question that we can address from the bottom-up angle or the top-down one. Okay, any questions about that before I jump into the bottom-up? Okay, and once again, these are the people. Merav Stern had crucial ideas here, as well as Stefano Riccanatesi and Matt Farrell, who helped address both of these questions. Okay, so what is it then from this bottom-up point of view about the way a network is connected that actually matters, so that it actually determines the dimensionality of the activity that it produces? Right, our first attack on this question, like many people's first attack in the modern era, is to just try a bunch of things, right? Let's just build a bunch of networks and compute their dimensionality. Okay, so by that, I really mean what I'm saying. You have a bunch of neurons. They have some sort of connectivity matrix, which we'll describe by W, right? And our question is going to be, what is it about that W that determines the dimensionality of the activity? Okay, some more ingredients. So every neuron in our simulation studies is going to be a spiking neuron, so we'll produce spikes. The model that we use to describe this is as a inhomogeneous Poisson process with a rate variable, rate sub I. Okay, so that simply means that the probability of producing a spike in a time interval is that rate times the length of the time interval. Okay, well, how does this W, how does this connectivity influence the rates and produce network-wide activity? The answer is that the rate for every neuron will be some baseline fixed value plus corrections to that fixed value that have to do with the way that the network itself is connected. So spike from neuron J comes through the connectivity matrix Wj to I and is filtered over time with some sort of filter that has a time constant, say, that looks like a synapse in the brain. This type of a model then that has a bunch of spikes that are coupled to one another is called a coupled point process or a Hawkes process. And this is the classical reference for this type of network model, coupled spiking network model. Who here, can somebody unmute if they have seen what's called a generalized linear model before, a generalized linear model with Poisson observation noise with a picture that looks like this, and this picture will ring many bells. Could any students who have seen this before just unmute really quickly and tell me where you've seen this? Or if you haven't, that's great, it's something new.
Yeah, something new for me.
Say it again, please.
Something new for me.
Something new. Okay, thanks, Gideon. Thanks so much for sharing that. That's great. Did I say your name right?
Yes.
Okay, fantastic. Okay, thanks very much. Yeah, so this is something new. And as you progress through theoretical neuroscience, Gideon, I promise you, you will see this picture with a dice here. And when you do, remember this talk, because when you see this picture with a dice, you'll probably be reading a paper that at least cites this one from Pillow and Paninsky in Nature and builds on the theory of Rob Cass and Emery Brown in this paper. And this is often used as a statistical model of neural activity, this type of thing where every neuron has a rate which is affected by other neurons in the network. This turns out to be a class of model which can be fit. As a matter of fact, the optimization problem for fitting a model like this to neural data is a convex problem. So it's become very popular in the field. People call this the GLM, and that in the context of neuroscience. And this is what they mean by that. So I want to emphasize that this type of model, for which I'm going to describe simulation style analysis, is also one that one can use in order to do statistical fitting to neural population data in order to infer these type of W's. That's an important connection. But for us today, we're using this as a dynamical model, right? I've got a bunch of neurons firing spikes. They do so at a rate that's influenced by the network. Okay, and again, let's remember my question. What is it about this connectivity that matters for the dimensionality of the activity that will be produced in a network like this? Okay. Now, what is it that we're going to measure for this dimensionality? There are two ways, two things that we can measure in terms of describing how far from independent neural activity is, right? So the first thing that we could measure is this average correlation, right? How far from independence, which would be a correlation of 0, are the neurons in the network? Okay, and as I'm simulating different neural networks here, making a bunch of networks in check, the one thing that I'm going to be varying is I'm going to be including more and more connections in this network. So that's this variable P that you see on this axis. I'm going to be making the networks more and more connected in my simulations. Okay, and as I do that, I see that this average correlation or average departure from independence goes higher and higher. It gets higher and higher as the networks get more connected. Okay, let me just start on this this corner here. When there's no connection probability, the network, all the neurons are firing independently of one another, and the average correlation is 0. Does that make sense? Does somebody say yes if that makes sense? Right, I hope so, right? They're not connected to one another, so how could they be influencing one another? And as you connect them together, this average correlation goes up, fine. However, remember there's this other thing that I can do with my covariance matrix, not just take the average of all of its entries, but take its eigenvalues as we just described, compute this weird dimensionality thing involving squaring the normalized eigenvalues and summing them up, and compute the dimensionality as a fraction of n, as a fraction of the largest possible dimensionality that you could get. And as you see here, as you make the network more and more connected, the mean correlation goes up, but the dimensionality goes down. This is also what you might expect. This result is not something that I would have expected. In many neural networks, and this is especially the case and really extremely the case in neural network models that describe the cortex, you may come across something in your studies called the balanced state, And that is something that happens for models of cortical activity, which have both excitation and inhibition and relatively strong connections. So there's strong excitation and inhibition, which is canceling one another out. For networks like that, the average correlation values, just like we see here, end up being pretty low. So let's look at the average range of average correlation values. In this range of simulations, we get up to 0.1. So on average, the amount of correlation that you'd see for a pair of neurons is 1/10. That is a very small amount of correlation. But look at the dimensionality values that co-occur with these very low values of correlation. Whoop, at a correlation factor of just 5% or so on average, the dimensionality is something like 0.1. So the number of dimensions compared to the number of neurons is 1 in 10, and this goes on further and further. So the message from this is one that says that, well, what is it about connectivity in these networks that is determining the network-wide dimension? There's something about the connectivity that is not, that can't be measured by the strength of these correlations. There are very low values of correlations, but these, and here's theoretical and experimental work that underpins that, but even when you have these very low values of correlation, at the same time, you can have very low values of dimensionality. There's a theory for this that I won't go through in detail, but I want to mention to you that this is related to this famous paper. So this is a paper from 2006 with the title "Weak Pairwise Correlations Imply a Strongly Correlated Network States in a Neural Population." And what this means, this is a more general statement of what we've shown on this previous slide. If you see that correlations are very low on average, and you're dealing with neural networks, That does not imply that the neurons are sufficiently independent of one another to have a rather high or full dimensionality. Quite the opposite. Neural networks tend to produce activity which has very low average correlations, but also has very low dimensionality. So these weak correlations on average can accumulate to have profound effects at the level of the population. That's an important thing to keep in mind, and that can be really surprising. So when you think you have an almost independent neural network, average correlations 5% or less, it could actually be highly constrained in its dimensionality. And again, that's interesting because many papers in the literature have emphasized that the average value of correlation is quite low. But this shows again that you don't know that that means that the dimensionality will be full. Quite the opposite. Okay. So back to our simulation. So we're studying these different networks. We're making bunches of different ones. The first thing that we saw was this interesting discrepancy between different ways of describing departures from independence. Low average correlations do not describe high dimensionality. But let's get into some more details here. So I want to describe in a little bit more detail some network simulations here to get us going. These networks are what's called Erdős-Renyi. That means they're completely random in terms of where the connections are in the network. The connection probability is my only variable, and for these simulations, all the cells will be excitatory. Okay. So we're back at this question, right? What is it about this connectivity matrix W that's actually important in terms of determining the dimensionality of neural activity? We've already seen a picture like this, right? As I make the network more and more connected, the relative dimensionality decreases. But if I construct this network in a different way, if I don't wire everything up randomly as for an Erdős-Renyi network, if instead I take another way of constructing a network, this is a typical thing that has hub nodes that's called scale free, I can take the same number of connections, 50% connectivity here, and have a dramatically different dimensionality. But if instead I wire my network in yet a third different way, making what's called a small world network, then suddenly, even though the structure is totally different from an Erdős-Renyi network, that doesn't seem to make any change to the dimensionality at all. So I think if there's a point that, the second point that I leave you with here, it's this. Our question, what is it about the way that a network is connected that matters for the dimensionality that it produces? The answer is it's more than just how many connections it has, but not all features of network connectivity matter in terms of producing dimension, in terms of impacting the dimensionality of neural activity. Why do we see this? Again, because I have two different neural networks that have totally different connectivity and they have the same dimension, but yet I can take a third example, which has connectivity that differs in a third way, and that has a dramatically different dimension. So this is the signature of an interesting problem, right? There is something, it's not everything, there's something that matters, and we want to figure out what it is. Okay, what is that something? So here's an idea. Let's think about what it is that neural networks or networks of any type are made of, right? Well, what are they made of if we zoom in? What are their ingredients? Well, one of their ingredients is connections between pairs of cells, right? And how much of that ingredient that you have is well described by a connection probability, okay, the only variable we've had so far. But there's more in terms of the ingredients of the network. If you look at link 2 connections, like this purple link 2 chain, or this blue, what's called a diverging connection, where you have one central node, there are all sorts of other patterns of connections that involve multiple cells at once. And I can enumerate how many of these I have in the network as well in terms of trying to describe what it's made of. These ingredients are called motifs. And I just want to give you a sense in this slide and these references that these motifs or connection subgraphs are under intensive study in experimental neuroscience as well. The question that we have here, though, as computational neuroscientists is, are these ingredients a good description? Are they enough to really solve the question of what it is in networks like this, actually determine whether the dimensionality will be high or low over and above their the connection probability itself? Okay, and these people, Yu was one of the early directors of this summer school, I think 2 years ago, maybe 3 years ago, and is an amazing mathematical neuroscientist, really incredible. And now he's at HCUST in Hong Kong. And these colleagues here, David Dahmen at ULIC and Stefano Recanatesi here in Seattle, they all give the answer yes. And Yu was the first one to develop the machinery together with a group of Pernice and Stefan Rotter in Germany to come to these type of answers. Okay, so they say these are the ingredients you need to concentrate on with an asterisk as usual. Okay, and for us, I think that this is an aha moment. I provided some of the papers, I think this one, to the group which contains all the relevant references. But this work of many people, I think, is significant. And the reason is, boy, if I would ask my colleague, Khan here, please tell me what it is about a W matrix that matters. It would be a challenge because that is n squared numbers, right? It's a humongous, very complicated object. And there's no clear way that you can write down in order to describe it. But if we said, well, don't give me all the details of the W, just break it down to its ingredients, right? And tell me how many connections does W have? And how many short paths does it contain of a few different types? And if we said that that's all you need to know about an entire network in order to predict its dimensionality, that is a useful summary of what it is that a network as a whole is made out of. It's a type of thing that you can measure in an experiment, as I indicated in some of those neuroscience papers. And it's also a feature of networks which evolves at the level at which local plasticity mechanisms, which tend to involve just pairs or perhaps localized ensembles of cells actually occur. Okay, so the machinery of how it is that you figure out that you can take a W matrix, which is a complicated thing, plug it into the type of mathematical model that I showed, and then reduce all the complexity of that W matrix just down to its path counts or these motif features is what is contained in these papers. And I'd be happy to talk about more. So I just want to mention a little. Yeah. Great. Yes, please, please.
Yeah, I have a question that I'm wondering, so when doing the research, why do they want to think about from a complicated W matrix to local plasticity?
Okay, that's a great question. So I think there's two aspects of this. So first of all, why do you want to just think about local mechanisms at all, right? And the answer is simply that is something that you can measure, right? If you need to just describe a neural network by how many motifs it has, for example, then you have to measure 16 numbers like this, and then that's it, right? So instead of a full connectivity matrix, which has n squared entries, which could be millions or billions of entries, you just measure how many of these different ingredients you have, and then you put that, and then that is it. That is all the information that you need in order to describe what the dimensionality is of the neural activity that will be produced. So why does anybody want that? And I think the answer is simply because it is tractable. It is a small list of numbers and it's a simplifying theory. You also asked about plasticity. And what is true is that it is activity in pairs of cells plus neuromodulatory effects, but really activities in pairs of cells that determines how the strength of those connections will change. So if you have a theory that says what really matters is on average across the network, how many of these different connections of small pairs of cells or linked pairs of cells like this you have in a neural network, then you say, well, this is what matters, and this is what is going to evolve as a network learns with the different plasticity mechanisms that it has. Does that make sense?
Yeah.
It's a great question, Pinja. Thank you so much for asking that. It also enables us to understand what some of the differences are in the simulations that we saw. So for example, to me, this was a giant mystery. Like, why is it that a network with Erdos-Renyi or random connectivity and a network with what's called small world connectivity have the same dimension, whereas this thing with these sort of hub neurons, a scale-free network, has dramatically lower dimensionality, right? So back to Pinja's point, if we looked at the whole W matrix, well, maybe there's some sort of complicated difference between these things, but what is it that actually matters? Well, what these theories do, right, is say you don't have to plug in the whole W matrix to predict the dimensionality. You instead can come up with just a short list of different statistics or features of that W, which again measure how many connections of what type that W actually occurs. How many of these little 2-cell chains does it have? How many of the blue diverging motifs? And then you plug those into the formula, and that gives you a good prediction or a good approximation for what the overall dimensionality will be. And that's powerful, not just because it's simple, but because you, or that's useful, not just because it's simple, but because then you can look at this F function, which relates all these different connection motifs and try and learn something about what it is that is important in a network in order to push its dimensionality up higher or down lower. And these things you can almost describe as importance factors. If you would take this F relationship and simplify it by linearizing it, these are just coefficients then that come in front of these motifs and tell you whether it is, when you have more of these type of chain motifs in a network, the dimensionality will go up or down. It would go up if that alpha is positive. Likewise, when you have more of these sort of diverging motifs, will that make the dimensionality go up or down? It would go down if that alpha is negative, and so on and so forth. And from these mathematical theories, we can actually compute what the alphas are, and it turns out that all of these are negative. And this gives us a way of explaining why it is that some networks will produce high dimensionality and some will produce low dimensionality. It turns out that the small world connectivity differs from a totally random connectivity in many ways, but not in any of the ways that affect the prevalence or the number of these different motifs that control dimensionality. However, when you go to make another type of network like the scale-free one, you end up adding more of these connectivity motifs, and we can establish that these connectivity motifs only decrease dimension. That must be why the network as a whole has a lower dimensionality. Okay, so here's my summary slide. I'm more or less on time, a little bit late. What are we doing, right? So we established that we care about the dimensionality of neural activity. And if we care about this, we'd like to know just what is it from first principles, or at least as best as we can, about neural connectivity that actually determines what the dimension is. And the answer that I described in this talk is that it is network motif. So it's not just how connected a neural network is. It's more than that, but it's not so much more than that that it's overwhelming to describe. Instead, we can use the methods that these people developed in order to break down this dimensionality, the origin of this dimensionality, into these different local features of circuits. Okay, so that is a general approach, and it's one that I've been personally privileged to be connected to. So that's why I chose to talk about this here. But this motif-based approach is not the only way. This is a summer school to approach this question of the link between connectivity and dimensionality. So I want to highlight another couple of approaches. This is a beautiful paper by more colleagues at École Normale Supérieure in Paris. It's called Linking Connectivity Dynamics and Computations in Low-Rank Recurrent Neural Networks, and Mastro Giuseppe is the first author. With the feature that these authors use is the low rank nature of W, and they predicted dimensionality from the rank of W. And that's also an example of a simplifying theory. I don't need to know everything about W. I need an estimate of its rank to figure out the dimensionality of neural activity. Here's another paper by Changcheng Huang and colleagues. And here she and her colleagues figured out how relative spatial and temporal timescales in other networks can be crucial in terms of producing spatial patterning in these networks that then determine the dimensionality of the activity in terms of the spatial extent of waves, for example. And yet a third approach deals more explicitly with nonlinearities in neural networks. And this is beautiful and very sophisticated work by Rainer Engelken that I'd like to recommend to you. Okay, so this is the end of part one. What I've offered in sum, again, is a definition of neural dimensionality, some statements on why we might care about it, and a tour of approaches that you might use in order to make a relationship or understand the origin of this dimensionality in terms of the connectivity among neurons. And I think we have about a 10-minute break or so after I went over by a few minutes. And then I think we'll discuss all this material again at the top of the hour. Does that sound right?
Yes, yes.
Okay, great. Well, thanks a lot. I look forward to picking up on this slide. I'll go get another coffee in 10 minutes. Is that okay?
Yep.
Great. All right. Thank you.
Actually, do everybody sort of understand the first part? If you have questions, you can prepare. We can ask when he's back.
Right. So maybe we can unshare the screen and then see everyone's
Yep.
face. It's,
Yeah, I see one from UD.
yeah, how is the first part of the talk? Is it clear? Any questions so far? Okay, there's a question on the chat room. Maybe we should wait until Eric comes back. Mm-hmm.
The last slide, recipe for dimension. You mean the equation to define the dimension, or?
Yeah, can you elaborate upon your question?
I think it's from Yueling.
Yeah. Can you turn on the microphone and the
Yes, Professor. Yes. It's the slide before the last slide, I think. It's a recipe for dimension. It's
from the
there's an
motif.
equation. Yes, it's the equations. I just want to see it clearly. Thank you.
Oh yeah. I think he's going to probably talk about that more in the second part. Details about how that's derived.
Okay, great. Thanks.
I think that it's the number of motifs, right? Different kinds of motif, chains or convergence and divergence that contributes to the dimensionality, with a coefficient that is negative, which can be computed and calculated analytically.
Yeah, I think actually it's interesting question from UD. Actually, it's really about how useful is this the measure of dimensionality, right? And how to use it, etc. I think that's really a great question.
Yeah, that's what I'm wondering, right? So this is like quite nice theoretical analysis and very good measurements. So I just like thinking overall, like what does because like brain science perhaps a lot of people say the goal of brain science is to understand behavior and the neural mechanism that generate behavior, right? So how does this simple measure of dimensionality related to the unsolved questions in behavior and brain functions? So yeah, that's
that's a great question.
Yeah, we should ask Eric for this.
Yeah, I guess he offers some heuristics of the good things about low dimensional versus high dimensional representations, right? But as a general question, I think this is a pretty hard one. We should ask him.
Right.
Oh, by the way, when possible, please turn on your camera. I see that some of you during the lecture. I mean, right now break is fine, right? But during the lecture, it would be nice to turn on the camera.
So you can see what I'm eating right now, say.
Is it good?
It's fine. Yeah, it is lunchtime here after all. But maybe half our students are probably eating.
Yeah, at the moment.
So I have also sent all the lecture slides to the Slack. On the Slack, you can take a look. Eric's slides and also Vijay's slides. I think that would be helpful.
Okay, I'm back. I can leave it to the course directors, however they want to.
Hi.
But for my part, I'm very happy to talk about anything that didn't make sense or that I can explain more of. I have some other backup slides and other things that I can use, anything that you wish.
Actually, Yudi, while you're away, Yudi had a interesting question. Yudi, would you want to ask that question?
Please.
Yeah, yeah. So my question is like, so how could you give us some more example of why dimensionality is related to behavior and brain functions? Because like I think like dimensionality, like you kind of like it's really nice theoretical analysis, and you kind of condense a lot of things into this one simple number. Right? So, but
Right.
like what really this number tell us about like how different brain functions and their underlying neural mechanisms. So that's what I'm wondering about.
Yeah, wow, that's a hard question. Okay, and a complete question. So that's very good. Well, let's go. Maybe I can try and start with maybe cheat by you know, going back to some of these examples that we looked at here. And so I guess we'll start with where we started. Let's just concentrate on this generalization thing. So this is a this is in a sense a question of how much information about an input you want to capture, right? So again, over here we had all of the different versions of Lewis tau here, right, in circles, right? And if these are spread out in many different dimensions, right, then I have many different axes or features that distinguish one version of Lewis tau for the next. But if I smash all my Lewis tau onto a plane, right, then we only have the two-dimensional description of Lewis tau, right? How much like a New Yorker is he today? How much like a Beijinger is he on the other axis? And so this is in a sense, a compromise, right? This is to say that you discard a lot of information and produce one of these low dimensional representations. What this tells us, I think, about function is again about this trade-off. In general, throwing away information sounds stupid. Why would I want to do that, right? The answer for why you might want to throw away information is because when you have a much lower dimensional representation of something, then you know what to expect, as I tried to indicate from just a couple of samples, right? If I'm trying to learn how to detect Lewis taus, if I see 3 examples of a Lewis, right? And then from that, I extrapolate to say, well, these 3 Lewises fill up a plane, right? What is going to happen when I get a new example of somebody and I don't know who they are, right? The first 3 times I saw Lewis, he shook my hand and said, "Hi, I'm Lewis," so I can know that these 3 red points are all Lewis. But let's say there's a new example that comes in, right? Now I'm at a conference and I see somebody far away. If their neural representation lies somewhere up here or somewhere up here, I'm going to have no idea whether it's Lewis or not, right? But if my brain has learned to compress all things that look like Lewis into this plane, then that new example will also fall somewhere here on this plane, and I can use whatever I learned before in order to figure out who it is. So that, I'm sure you know, you have a sophisticated background, but that is what people mean by generalization. And so if what the brain is doing is taking all these different very high dimensional inputs and somehow squashing them into low dimensional sets, that might be telling us that what the brain is valuing in terms of its computation is not remembering all the details, right? But compressing things down to the essence so that all the unnecessary details are thrown away. And what remains of an image is what really matters in order to categorize it usefully. And so that is just, you know, a question of the brain's function. If that's the case, that would be a very interesting, but in a sense, very understandable and logical way for it to represent information. But it's a completely different perspective than the picture that you get from machine learning. It's a different perspective than the classical pictures that you get from thinking about high dimensional representation in the cerebellum, for example, which were ideas I believe that Marr and others were associated with. It's a very different idea than you get from the work of Alex Kaiko-Gaich, yet another colleague at École Normale Supérieure. Here's how to spell her name: C-A-Y-C-O space G-A-J-I-C, Kaiko-Gaich, Alex, C-A-Y-C-O space G-A-J-I-C, or the work of Ashok Litwin Kumar, last name Litwin-Kumar, at Columbia. And what both of these people did in articles in Neuron and Nature Communication in 2017 is to argue that there are structures in the brain that all produce this type of high dimensional representation, again, for the purpose of producing, as we described, easy separation of different classes. So I think when we look in the brain or in a brain region and we see whether it's high or low, that tells us something about whether I'm really working to separate patterns and prepare to make categories, or whether I'm preparing to generalize and extend what I learn from a few examples to new situations. And the last comment that I make on this, which I think is extremely interesting, and I have a couple slides I can share on this. If UD will go back to you, you can ask for them or not. Which is, okay, let's say I've decided I want to compress Lewis into 2 dimensions, right? Because I don't care about all that detail about Lewis. I just want to be able to meet him 3 times. And then when he comes to meet me by the clock and bell tower and my wife, last time we were in Beijing, I'll be able to actually recognize him. Okay, well, then the question is, what features? What are the features that I retain? What are the features that actually matter, right? What co-varies with these neural responses in this low dimensional plane? And it turns out, so what is thrown away and what is it that you capture? That has to do with what categories you want to be able to continue to distinguish. And that can get extremely interesting when you find low dimensional representations in the brain to try to figure out what exactly is kept. Because that tells you what the categories are beyond just image recognition or colleague 1 versus colleague 2 that the brain is trying to remember. And if you wish, I can show a specific example of that. Okay, was that okay, Yudi, or was that just sort of saying things that I already said and you understood and was wasting your time?
Thanks so much. That was very insightful answers. Yeah, I'll look at the paper. What was the second paper you mentioned? I think the first paper you show has posted on the chat, but I missed the second paper.
Oh yeah, let me just do that. I forgot about the chat. Okay, let me see if I can open the chat. So Alex Kaiko-Gaich. So the Alex Kaiko-Gaich paper went up there, great. And then I'll just get the other paper, Neuron, Litwin, Kumar. Yeah, I think Yudi, your comment about a trade-off. Sorry, I'm seeing this now. What those amusing things here. Okay, sorry, I'm not looking at the chat. So and I'm glad because Louis would have made me laugh. But let me get this. Yeah, so here's the paper of Litwin Kumar. And yeah, and there, that's in the chat. And again, I think I was clear in saying this, but what reality is going to be, right? So you'll see some papers talking about high dimensional representations, like the 2 papers in the chat talk about high dimensional representations. Here, I guess I'm just talking with a simple cartoon about low dimensional representations. But if you look in this paper or others that cite this paper, this is Cunningham and Yu in Nature Reviews Neuroscience in 2014, you'll see lots of papers that talk about low dimensional representations. It's not true that this is right and that's wrong, or that's wrong and this is right. What is true almost surely is that different brain areas are designed to do different functions, and at different stages through learning you'll see either high or low dimensional representations. And that's actually in part 2 of my talk, or of my lectures, I guess, and next week or whatever, I'll talk explicitly about that. And it'll be quite interesting. We'll see over stages of learning, or even as we look at deep neural networks, when we look at early layers versus deep layers, we see going from high dimensional representations, which are good for forming general categories, to low dimensional representations, which form these efficient type of descriptions. Okay, anyway, thanks for a good question. And great, okay. I'm happy to take another question, or I can show an example. Maybe I should just take other questions. Let me just be quiet. And a good question is like, I didn't understand what you said, or that slide was weird or something. I mean, these don't have to, UD asked a very very nice question. But other nice questions are just like, tell me what was on that slide, or give me some more background, or you skip too much stuff. Anything is good.
So there's another question previously on the relationship between dimensionality and the motif, that equation, I think. Maybe you can elaborate upon that as well.
Yeah, exactly. Where does that actually
ask this question? Yeah.
Yeah, that's great. Let me just get up the right slides for that. I'll reshare my screen just 1 second as soon as I get the right slides. Yeah, okay, so this is it. Okay, so. Okay, great. So here we go again.
Can you share the screen? Or
yeah, I just had to get the right slide up, so it made sense. Okay, there we go. So here is the slide. This is a hidden one. Okay, so let's just do this. And again, we'll cite the appropriate scholars as we go along here. This is work of the community, as all of science is, as we often forget. Okay, here we go. All right, so remember our model, right? And the reason I'm telling you about the model is because it has the explicit entry of the W, right? And we had a nice, I believe it was Pigeon, if I am saying her name right, I'm sorry if I'm getting it wrong, already asked us about the W matrix very nicely. Okay, so this W matrix is a big complicated thing that tells us how cell J is connected to cell I. Okay, now we also remember this dimensionality formula, which is related to these squared eigenvalues, squared normalized eigenvalues. But many of us will remember that eigenvalues, or this is a sum of squared eigenvalues, that the sum of eigenvalues is the trace of a matrix, and the sum of eigenvalues squared will be this will be the trace of that matrix itself squared. So what's very nice is that this dimension quantity, which looks a little bit weird, can be related back to this covariance matrix with some simple trace type of operations. Okay, now why is that nice? The reason that that's nice is that Sejnowski, what's the reason why that is nice is that I want that slide, yeah. The reason why that's nice is that this covariance matrix itself can be written explicitly in terms of the W. I've left out some terms here, but the basic idea is you take this original complicated W, and you take the difference with the identity matrix, and you take some inverses and things, and then you get an explicit expression for what the covariance matrix is. Let's remember what we're doing. We want dimensionality. We want to know how dimensionality depends on W. Dimensionality is directly related to the covariance matrix, and here we have some sort of expression involving matrix inverses that relates the covariance matrix back to W. So this should enable us to relate the dimensionality to W by plugging in. The problem is, if you just did that immediately, you would end up with a trace of some sort of matrix inverse of W, and you could shove it in, and you would get the exact right answer in terms of what the dimensionality is. But what you wouldn't have is an understanding of what's going on, right? It's sort of just like a simulation. Give me a complicated W, and I'll tell you what the dimensionality is by plugging it into something that I can't understand, right? The progress comes from reading the work of Pernice and Rotter, who are in Germany, reading the work of Truesdale and Josic, who are in Texas. And what these two people, these two groups realized is that these matrix inverses can be expanded in power series, right? Just like 1 minus x to -1 has a power series expansion. Basically 1 minus a matrix inverse also has a power series expansion. And if you do that, these matrix inverses, which are very difficult to interpret, become sums of powers of the matrices. And when you multiply these two things together, you'll get a bunch of terms. The first term will look like the identity matrix. The next type of term would look like just one copy of W, and then you'll have all powers of W and W transpose. Okay, stay with me now. So what is going on? Then you take these things and you shove them into these trace operations. And what you're doing is basically averaging over, you're basically averaging over the matrix and taking this trace type of operation. It's something like a sum. But what is it that you're averaging over? This is the final point. Well, first you're averaging, when you plug this in, something like the identity. That wasn't very exciting. The next thing that you're averaging is something like the raw connectivity matrix, W. Okay, what is the raw connectivity matrix when you average it? Well, that's going to have to do with just the entries of one copy of the connectivity matrix. And the average of that is something like the average strength or the average probability that you have a connection in the network. What about some of these other matrix products? What do they correspond to? Well, it turns out that, let's see here, just 1 second here, I'm going to unhide this slide. Work. Skip, whoops, unskip slide. Let's try and play this. Let's see if this works. Okay. Okay. Well, that's exciting. Okay, so just to continue to this, so it turns out that non-zero values of matrix products. Okay, here we go. Non-zero values of matrix products, like this green term, map onto the average, excuse me, of this green term maps onto averaging quantities that correspond to link 2 connections. What am I talking about? You have one W, you multiply it by another W. And when you do that matrix multiplication, as all of us remember, you're combining one entry from the first matrix with another entry from the second matrix and summing them up, right? And it turns out that when you do that sum, right, one entry from the first matrix W corresponds to one connection. Another entry from the other matrix corresponds to another connection. And when you are taking the product of those terms, you are measuring how strong connections, the product of these connections are in the network itself, which is like measuring how many motifs that look like this are actually in the network. The next term involves 3 copies of W, and that's going to involve connections that involve 3 of these arrows here, and so on and so forth. So the bottom line is that this dimensionality formula ends up being writable in terms of a power series of different products of this matrix W, and that ends up being like a power series, and every one of those terms corresponds to a motif which is larger and larger in its length. So our final formula for dimensionality ends up looking like a sum of a bunch of strengths or probabilities of these different motifs. And then the final step to show that you need only small or low order motifs is to do some rearranging of these sums and resumming these power series. And this is what Yu Hu, who's now again at HCOST, figured out how to do. And to show that the higher order motifs can be expressed as functions of the lower order motifs. So you can end up with very efficient truncations of these sums in the end, which involve only a few motifs at a time. Okay, that was an attempt. Now I'm ready for questions. What was unclear or weird or seemed wrong in what I tried to say? Anything? Did that help at all?
Yeah, there's the student. Jiajun, is it Jiajun? I think it's another person, right? It's from the.
Yeah, I'm from EW.
Oh, hi, Jiajun. Nice to see you. Hi.
Hi, yeah, I'm a grad at UW and then majoring in applied math also.
Wonderful.
Actually, yeah, I was just typing a question in and then hi professor, and then may I ask, is there like a specific form of the small motifs look like compared to the W, like the matrix? Like for example, how can we detect those small motifs based on the W we have, the complex
Right,
W we
yeah.
have?
Yeah, you can. So basically, so let's talk about, so the first motif, right? Or the first feature of a graph, sorry, let's see if this is working. Whoops. 1 second. All right. Just a moment. That was sad. All right. Good. So the first the first feature that you see, right, is this blue thing, which is just W, right? And so W corresponds to just paths of length 1, right? And it turns out you have to think about, so one has to think, and I have to get lost, and then other smart people figure it out and tell me what the answer is. Basically, that's the way my research life seems to work. You have to do these trace kind of operations, and it's not that obvious. You have to rewrite the trace expression in terms of really averages over all the entries in the matrix in some sensible way. But when you figure out how to do that, you see that anything involving just this W matrix is going to involve just one connection at a time, right? That's what I was trying to say. And anything involving like this WW squared, or let's go to this one, this 3-link thing corresponds to the statistic at non-zero entries, or entries in this matrix correspond to all the strength or the sum strength of motifs that look like this. Very specifically, this is a matrix, right? So it has rows I and columns J, and the ijth entry of this matrix corresponds to the weighted number of link 3 paths that go to that terminate in neurons I and J and go through any intermediate neuron K, right? So that is when you just do the matrix multiplication, you can see again what non-zero entries in these matrices Zhaijun correspond to. So then the last thing that you have to do is figure out, again, what are these trace type of averaging entries, or what are the averaging type of operations that you're doing on these matrix products? And those really end up corresponding to just counting or taking the weighted average across all entries ij of these matrix products. And so in the end, then, these formulas end up looking like a sum of matrix-wide averages of these matrix products, which is exactly which corresponds, as we were saying, to the average strength or the average presence of these types of connection structures in the networks as a whole. So to answer your question very directly, if you want to compute the important motif statistics, or what we were talking about as the ingredients in the networks, if I dare bring up this slide again, right? If you want to compute your ingredients list, all you have to do is take a given connectivity matrix W and multiply it by itself, or by itself with its transpose, some number of times, and take the average, and you'll get the ingredients that you need. And so that's useful, again, if somebody gives you a W matrix, and that's important for some of the theoretical calculations. But if you come at it from the other side, which is like, what is the brain doing? Like, what is there? Most of the data that we have about a brain comes from doing experiments like that, which I'm indicating here, a famous 2005 paper here. And there they get some number of electrodes, and you patch some number of cells, here 4, and literally you test, like stimulate neuron 1, is there a response in neuron 2? or not, right? That tells you whether there's an edge here or not. And then you do that for all, I guess, of the 12 different combinations here. And then you say, well, what type of connections did I have? So this thing, which is called multiple patch clamp analysis, is the primary way in which we can get detailed synaptic information about connectivity in a network. So you can take it, if you're a biologist, you would take it from this level, which is to say, look, these ingredients are the only thing I can measure. I'm never going to have a W. Never. Don't have it. Who has the million entries with all possible pairwise things, even for 1,000 cells, or for a million neuron thing? Who has the 10 to the 12th different combinations, the trillion pairs of synaptic connections? Nobody, right? And so all you have is these ingredients, and you need to, and that's all you can measure. And then from the theorist, again, they say, well, I just take these simple matrix products. I want to point out that an exception to the rule here, I'm sort of complaining about from an experimental point of view, you never have a W. Many of you know about connectomics research, right? And there are wonderful centers for this all over the world. But the microns group, let me actually release, not release, just share the link here in the chat. One moment. There we go. Okay, so there's a website now in the chat, and many of you have probably seen this. So there are groups now that actually are measuring the entire W in small chunks of brain, and that will, you know, in all of our careers, including the younger people's careers, but even in mine, that will become the norm. And so we will be getting more and more detailed information in the future. But as for now, it really is just these small sort of localized circuits that we can measure. And it may be and one of the papers that I put a reference to, so this thing by Daman and Rakanatesi that is now being worked on, is on the archive. You know, what these, sorry, let me just put it a different way. It may be that what we want to do in these very large connectomics data sets actually is to process them to extract their simpler components in the end, because even if we can measure everything, that doesn't mean that we'll be able to come up with an understanding of how all these pieces work together. Okay, anyway, thanks for a great question. And I'm really happy to take any more questions or clarify on that answer any way I can.
Oh, can I ask a question?
Anything.
Yeah, so as you have mentioned this result about this relationship between the motifs and this dimensionality of the network, and all your result and framework has built up on this Hawk's process model. I mean, these dynamics of Hawk's process. Yeah, so I'm wondering if we can switch this dynamics or switch this model to some other models where this result still remains similar, or is this framework can covers more generalized version?
Yeah, thank you so much. And yeah, I think overall I need to be more humble. Like Kai beautifully pointed out, and I tried to put an asterisk somewhere about this being a linear theory, but that's really important, right? This kind of description of cells as interacting Poisson processes is pretty reasonable, I think, but this red thing about the linearity is not right necessarily, right? And so in general, you would have some sort of a nonlinear function here, like you would in an artificial neural network that's doing any significant computation as well. And here we've just linearized that and built a theory for that. So I think there are a couple of answers. So if this, just like always, if this linearization is a reasonable description of the nonlinear system, then the approximation that you come to through this motif theory will continue to work. And so, for example, you can replace this model with a integrate and fire neuron network where every neuron is receiving independent Wiener process or white noise and couple that together. And you will see that when you do the linearization and map it back onto a model of this type and count the motifs, you get a prediction for correlations or dimensionality, which is close to that in the full nonlinear simulations. But if you have a very bistable system, right, where the activity is sometimes very low, so you might want to linearize here, and sometimes very high, where you might want to linearize there, and you jump back and forth, then this linearization will be a bad approximation, right? And what you would need to do then is account for different discrete states that the network might be in, right? And do a linearization around each of those states. But then in combining together the shifts between those states, that is something that that is a type of transition. Anyway, those type of dynamics, the transition between states, this theory would completely fail to explain. And so failure. And I guess what I should have done is say more clearly on this slide that all of these other, well, I'm not sure about this approach, but certainly this approach here is nonlinear. It's a linear stability analysis, but the things that it predicts map onto the nonlinear cases. And also, I want to put another paper in the chat. I'm going to do that right now. So Williamson, Doron, 2016 loss. Good. So here's another really nice paper. In the chat now, and that's a paper that, if you look at the models in this paper, that explicitly deals with these types of bistable or multi-stable states, where the dimensionality is controlled by the number of different multi-stable states, and therefore our motif-based method, I think, would probably fail in that approach as well. So I think here's the best thing to take. So thank you, Kai, so much for your question. And I think really the thing to take home from this is, if you believe, right, so we have to question the model, and Kai was very smart to do that. If you believe that this type of a description is reasonable for your neural system, it's roughly at a stationary operating point, and you're interested in fluctuations around that, then this will work, and this probably will be general enough to answer your question. But if your system is moving between different operating points and is really importantly exploring its nonlinear dynamics, then this will completely fail. Then you should be like, well, that was the nice sort of fun, 15 minutes or 20 minutes of talking about this with Eric at the C-Neuro, but I'm going to throw this approach in the trash, at least until somebody really deals with a fully nonlinear case. There's one other point, which is that you might, as a theorist, you might know that what people always do is they linearize a model and study that, and then sometimes you go for a power series expansion, or you might expand this nonlinearity as a power series. So first you'd say it's linear, then you'd say there's a quadratic correction to that, then you'd say there's a third order correction to that, and you would make corrections to your model at each of those orders. That is something that has also been done. So this is Bice, Walker, Ploss. So here's a paper that takes that approach and succeeds. Gabriel Ocker and Michael Bice being the key authors there. But I would still say, again, Kai, that that gives successively better approximations around this point. But if you have fully nonlinear dynamics, I think it still will not work. So this is a way, again, of dealing with the type of fluctuations you see around a stable operating point in the system. And if that operating point is changing all over the place, you will not be able to capture the structure of those transitions with this type of a description, and you need other harder and more interesting, perhaps, or mathematical approaches in that case. It's a very good question. Okay, others, or Kai, any clarifications?
I think maybe we can, if you have more materials, maybe we can go on a little bit more.
Sure. Okay, great. So let's see what would be.
We have about, I guess, 20 minutes.
Oh, okay. I was slated to go till 9:45 here. Let's see. So
I mean, I guess I guess the rest of the time was for questions. I guess we did it, yeah, in advance.
Okay, that's great. Let me just see if there's one more thing that.
So I have a quick questions about this linear response theory. Is there, given that we have so much experimental data these days, is there a way we can actually, at least some empirical way, to differentiate these models? For example, those.
Yeah.
Right.
Yeah, I think so. I like that very much, and I think let me put in the I think we will go back to this same slide again. So share the screen on this. Okay. So this, I think, comes to do with the fact that these types of models, right, which we described as you said you would, I said you would see these in the literature often as Poisson generalized linear models. These are the types of things that can be statistically fit to neural data. So let me put a link to a modern paper that does that in the chat. Great. So this is a really nice modern review paper. Oops. I'll just put the PubMed citation. Chat. Okay. And then let me get up the paper itself and share that so we can talk through it. So nice to have a lot of time for discussion. You plan this well. All right. Okay, this will more or less do it. Okay, so here we go. So here's a slide from that same paper. Here we go. Okay, so you'll see this Poisson generalized linear model, right? And the basic idea is, forget this region thing, we can ignore this. Let's just say this is a bunch of spiking neurons, right? You're trying to predict activity of neuron number J, right? And here's the model. It's the same model that we were just talking about. You have a bunch of spikes, and each one of these spikes passes through some filter as a function of time, and then passes through a, let's just say there's a temporal filter and then a weight on that temporal filter, which are separate, right? So this would be like the Wij terms. And these things are added together with a Wij term. They're passed through a nonlinearity. Here, that's the same nonlinearity we were talking about linearizing. And then that is used to predict the firing rate in neuron number J. Okay, so why am I showing you this again? It's like, excuse me, you've told me this 3 times. The answer is, what is this paper about? This paper has absolutely nothing to do, this is a great paper, it's a really good paper, but this is the paper in the literature that is not about modeling neural circuits as dynamical systems. This is the literature that's about really good statistical models to describe neural data that are directly fittable to neural data. So as I tried to explain, this type of a model, even with a carefully chosen nonlinearity, right, can be directly fit to neural data in a convex manner. Okay, so I think, Sean, that this would be the approach that I would recommend taking, would be to say, well, give me a brain area, fit a model like this, right? See how well that describes the data, right? By doing something like maybe evaluating the likelihood of the data, of held out data according to the model or something like that. But anyway, do that with a real nonlinearity. Then do that with a linear model of this type, right? Right, so get rid of the nonlinearity and then refit the model. That can be done. That's also convex. That's easier than having a particular form of, not, well, it doesn't really matter. They're both easy fitting problems, but the second one is probably almost an analytical problem. But anyway, do it analytically solvable optimization problem. But anyway, do it both ways and ask how much worse is the nonlinear model in terms of describing the data and predicting the activity of neuron J. And I bet what would happen would be that if you have one of these cases that I described, where you're moving between different operating points and linearization is hopeless, I bet it would really make a big difference whether you include a nonlinearity or not in this fitting. And if you don't, in other words, if you're operating roughly around a stationary point, then you really will need then you really will succeed, in the sense that the linear model and the nonlinear model will be pretty close in terms of being able to reproduce the activity be able to predict the activity in one neuron from the activity in others. Does that make any sense?
Yeah. I got
That
it.
might work.
Thanks a lot.
Yeah, and then something that's in between, which I think is worth saying. I mean, we have to. Yeah, I think this is really useful because, I don't know, maybe I had too much coffee, but I made a mistake in my lecture, which was to be very enthusiastic about this modeling approach. And then Kai rescued us by really demanding that we are serious about what the modeling assumptions are. So that was very good. So thank you again, Kai. Anyway, so I just want to acknowledge that and thank that again. But you know, I think there will be cases which look like the following that I'm about to show you. Let's see here. So I need to get this. Let's see, will this do it? Right. So this is maybe work for the future. For the next generation or the next set of work in this area. All right, so now I'm going to stop my share and reshare a different example. Okay, so here's a paper from our group, and here's what Daman and Rakanatesi do. So they take this. This is high dimensional neural pixels data. Doesn't matter. This is electrophysiological data from hundreds of neurons at once, right? As is becoming actually publicly available. One awesome place, I cannot resist saying this. Many of you know this, but brainmap.org. Okay, so we'll just put it here. Well, that's slowly loading. So I'll share in the link a place where all kinds of data like this has been made publicly available from the Allen Institute. You probably know about this, but there are other places also, the laboratory of Nick Steinmetz, for example, now the University of Washington, that share data of these type. But the point is, you've got lots of data like this, lots of neurons at once. Okay, now try to describe this by the neural data, which is like, let's just say 200 variables at once, 200 variables, this time step, 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 as the emissions or observations from a hidden Markov model, which is a model that has discrete states, and then fit that hidden Markov model. using the software of Scott Linderman, Linderman, Linderman. SSM package. So here's Linderman. It stands for a package called SSM available on GitHub, which is an amazing package, beautifully curated, which does a lot of these type of spike train analyses with latent variable models like HMM models automatically. So thank you, Scott Linderman, leading the way on that. Anyway, so thank you, Scott Linderman, apply that to these data and try to identify discrete states. And when you do that, the discrete states in this case are white and green and blue. Notice you get multiple discrete states. It is not true that everything is all one state, right? And so the type of thing that alerts you to is that this linear response theory might fail, and you should at least separate out the data into these different states. Separately try to do things like predict correlations, then put all the data together with the transitions and look at correlations and dimensionality and see if you get a different answer. And so in this case, it didn't make a difference whether we parsed it separately, and we felt like the linear response theory was reasonable when it was applied to all the data. But in other cases, like those I was talking about with Kai, and the rest of you, it probably would make a difference. But this is at least a tool to separate out the data into different discrete time intervals, and in which this type of a linear theory may be valid. But if it is not valid in the end for the neural dynamics that you have, or if you want to understand the full set of transitions, that is completely possible that will be the case. You really will need a very different theory, and I do not know whether this localized motif-based approach will work, or whether there's something really different that we will need. It'll be cool to find out. Great. Okay. I'm happy to keep going or call it a night, just as you all wish.
Yeah, I guess if we have more questions, or
yeah, please. Questions are the best. They've all been very helpful questions. Okay, there's one more thing I can share if people want to hear it, or we could. I'm not quite sure when we were supposed to stop, or I'm also happy to stop and collapse depending on what the time is for everybody. All right.
I guess we can.
Yeah, please send. What should I do? Up to you.
Yeah, I guess we can. I guess according to the schedule, I have 2 more minutes. Maybe we can.
Oh, 2 more minutes. Okay. Okay, I didn't realize, just 2 more minutes. Okay, so. Yeah. Okay. Let me try to just offer a slightly better or more full description of this slide, which I skipped past. Okay, so there were 2 things that I was talking about, right? Like most of my talk was about dimensionality, right? But then every now and then I mentioned this average correlation coefficient, right? And Lewis Tao will remember that back when we were all in New York 15 years ago or something like this average correlation thing really was something that many people, these are classic papers, were concentrating on in terms of trying to talk about whether the brain is independent or not. It makes sense, right? On average, measure the correlations. And if they're close to 0, it sounds like on average, the brain is correlated. And for certain quantities that you might want to compute that have to do with linear readouts of populations, this average correlation coefficient actually is the key quantity. But the thing that I wanted to point out, and this may be my last remark, is that when you look at these trace type of expressions, which as we saw enter into the expression for dimensionality, there's actually a third way of rewriting the expression, right, for dimensionality. We saw it in terms of the sum of the eigenvalues, the normalized eigenvalues squared. We saw it in terms of the trace of C and C squared, which is the same thing. But if you evaluate those trace terms, you find out that it is a function, a very simple function that's in these papers of the mean correlation and also the variance from one neuron to the next in the correlations. And that is something that I did not appreciate for years of thinking about independence in the brain. And it turns out, as you'll see in these papers, that these two factors, the mean of the correlations and the variance or variability, if Lewis is positively correlated with me, but Chan and I are negatively correlated, then there's going to be a variance in the CIJ between different pairs of neurons, and that contributes equally to dimensionality. And so a few of you may have, all of the faculty, and maybe a few of you who are newer may have studied these things called balanced networks. I already mentioned them once. But it turns out that in these balanced networks, you can see that both these mean and the variance of correlations are small, but when you plug them together into this formula, you see that even the small variability or variance in the correlations that remains is enough to control the dimensionality. So that's a piece that was missing. These are kind of mysterious numbers like dimensionality. That sounds very complicated and very sophisticated. I don't know, to me, it sounds sophisticated, I guess. But the important thing is to remember that this comes down to very simple, measurable features of the correlation or covariance matrix. And when you think about large networks, the even very, very small values of these statistics can have a profound influence on how the network as a whole is operating. Okay, that's one bit I didn't get to explain. I will see some of you, or maybe it's a whole new set of colleagues at 9 o'clock in the morning, Seattle time, which who knows what time that is in everybody's time zone. But thanks a lot for the great questions and for especially thinking critically and challenging some of these points. That's actual science as opposed to not doing science and mathematics. So thank you. It's a wonderful group. Should we?
I'm sorry.
Yep.
Okay.
Hi, Professor.
Yeah.
Yeah, sorry to ask this question at this last time. But,
Yeah, no.
I'm wondering that, so why to think the stimulus as low dimensional? Because is it really low dimensional? For example, when you see things using our eyes, maybe the picture in front of us is thousands of
Exactly.
pixels, and when we're hearing, maybe it not only has information of frequency, but also has information of time, space, etc. So is it a direct or is it a direct thinking that to see the stimulus as low dimensional? What if we see it as high dimensional?
Yeah,
And
no,
if we
yeah,
see
another
it
great
as high dimensional, what's the way out?
Great, another very, very good question. And I think that you're absolutely right. So it's absolutely true that most stimuli in the world are high dimensional, period. And with the question though that we want to answer, and we'll do this explicitly when I come back in a week for part 2, is when you take a high dimensional input, like you say, like a pixel map, and you put it into a neural network, what happens, right? And is that high dimensional input reprocessed to a low dimensional representation? So Lewis is high dimensional as a pixel map, but what does my brain do to Lewis, right? Does Lewis create a low, does my brain make something high dimensional into something low dimensional? And we'll see actually when we train recurrent neural networks to do different tasks in the next lecture, that seems to be what neural networks do. So again, it's what does the brain do, not do with a high dimensional stimulus. That's the most important question for us. That's a very good question.
Yeah, thank you.
Great, super good, thank you. Okay, I'll hang up, see everybody, and thank you again for the opportunity. It's really
Thank
valuable.
you.
For me. Thank you. Bye-bye.
Thanks, Eric.
Thank you.
Thank you,
so
Eric.
much. Thank you. It's my privilege. Good night.
Good night. All right, we should maybe chat a few minutes.
So
Yeah.
I will stop the recording now.
Yeah.
Like,
You can stop
is there some
recording
place
now.
I can upload the recording, or like I can wait for later?
Well, I think you can, because you're in Beijing, right?
Mhm.
You can always give it to THVI, go to the headquarters.
I
And
see.
give
Yeah.
them the
Yeah. Okay.
yeah.
Personal